I'm Nadia Thompson, and today I'm going to be talking to you about the Kras protein, which originates from the Kras gene, or Kras, but I call it Kras. I'm going to tell you about the mutation of the glycine being replaced by aspartic acid, or cysteine, or as they call them, G12D or G12C. So the normal function of the Kras gene provides instructions for making the protein Kras. This protein relays signals from outside the cell to the cell's nucleus. These signals instruct the cell to grow and divide, or to mature and take on specialized functions. The Kras protein acts as a switch that is turned on and off by the GTP and GDP molecules. To transmit signals, the protein must be turned on by attaching to a molecule of GTP and is turned off when it converts the GTP molecule to the GDP molecule. And when this happens, it does not relay signals to the nucleus. I would identify this function as a receptor because it signals the nucleus to provide cells specific functions. The location of this protein is relatively found at the inner surface of the plasma membrane. They are also mainly found in animal cells, and I'll explain why in a little bit. The type of change with this mutation is with the mutation the protein becomes more active. I say this because the G12C mutation occurs, it amplifies the signals that control the dividing and the growth of the cell. And when this happens, it can lead to tumors, which leads to cancers in the body. So I have some 3D models here, and I used, for the first, I had to do 2 3D models. For the first one, I used this picture as a reference, which is a wild type Kras signaling. My 3D model is right here. So basically, this is the wild type Kras gene itself. All of these little ones right here are basically the small mutations that are within that signaling. These little yellow ones right here show the signals moving from one mutation to the next and eventually hits the nucleus at the bottom. These little holes that are in this nucleus are the nucleus pores. Those signals go through the nucleus pore and signal it straight to the nucleus to show the cell how to basically grow or divide, and it keeps it under control so that the growth and division does not go out of control and create these tumors. On the other side, I have the mutation, which is the G12C mutation, which is this picture I referenced. And it basically looks like this. So definitely you can see a drastic change in the mutation. It takes a lot of the little tiny mutations that were around the first one out and only replaces it with 3 small, almost medium-sized mutations. This is the mutant G12C Kras. And these signals, it just shows, as you can see from the first one, there is only one signal to the next. This one has about 3 signals to the next mutation. So as you can see, these signals go out of control, and the division of the cells begins to rapidly pick up, and the growth of the cells begins to rapidly pick up as well, causing these tumors in your body to create the cancer that some people can get. All right. So the change in the structure because of the mutation, as I said, mutations are taken out of the structure and the signals become more hectic, and more signals pass through from the protein. There are 2 main functional groups in the original amino acid side chain that are from the original wild type Kras. These function groups include, These functional groups in the original amino acid side chain include amino groups and carbonyl groups. The properties of the original amino acid side chain, both of the functional groups present in this amino acid side chain are hydrophilic, meaning they are water soluble. They are both polar, so this amino acid is hydrophilic and polar. This amino acid is small compared to other amino acids, and this is because it is also considered a simple amino acid. The bond types to participate in the original amino acid is it can participate in hydrogen bonding because of the hydrogen attached to the oxygen in the chain, and permanent dipole moment because of the electronegative atoms attached to less electronegative atoms. Properties of the new mutated amino acid is that cysteine is the one that replaces it. So cysteine has 3 functional groups in its chain, which are amino groups, carboxyl groups, and sulfihydryl groups. It is a polar amino acid and is not charged. It is smaller than other essential amino acids because it is a semi-essential amino acid, but it is bigger than simple amino acids such as the glycine. So comparing these bond types from the original to the mutated one, the mutated amino acid has a much stronger bond because this amino acid is much bigger, so it deals with peptide bonds, which is a covalent bond, and covalent bonds are much stronger than the hydrogen bond dealing with the original amino acid. So lastly, how does the mutation affect the functions in the cell, in the protein itself, and the body? This mutation affects the protein by sending more signals than needed through the cell, and it changes the shape of the protein as well, as you can see in my 3D models. It affects the cell by allowing it to grow and divide at a rapid rate, and this affects the organism because it causes tumor, which leads to cancer with the organism. Many types of cancers that are associated with this protein are lung and pancreatic cancer, which in some cases can turn severe. So there's my presentation on the Kras gene and Kras protein and the mutation that comes with it. Thank you.